Hello, my name is Joshua Mugabi. I welcome you to Enzymes. Bingo. So without wasting any of our time, let's go directly to our quiz. The active site of an enzyme is not a fixed shape. It can change slightly to better fit the shape of the substrate when forming the enzyme-substrate complex. A. Bioprocessing. B. Lipolytic. C. Induced fit theory. D. Specific. And the answer is C. Induced fit theory. The minimum amount of energy needed to start a chemical reaction. This varies from reaction to reaction. A. Maltase. B. Energy of activation. C. RNA polymerase. D. Erepsin. And the answer is B. Energy of activation. Enzyme which is fixed to an inert material by chemical or physical means, e.g., trapped in beads or gel, so that it will react with but not mix with its substrate. This makes it easy to recover the enzyme from the product, and it is reusable. A. Lysosome. B. Enzyme-substrate complex. C. Immobilized enzyme. D. Lytic enzyme. And the answer is C. Immobilized enzyme. The position on an enzyme where the substrate is attached. A. Active site. B. Specific. C. Bioreactor. D. Catalyst. And the answer is A. Active site. Enzymes produced by the salivary glands, the pancreas, and the small intestine. It converts starch to maltose during digestion. Its optimum pH is slightly basic. A. Protease. B. Hydrolase. C. Amylase. D. Lactase. And the answer is C. Amylase. Any enzyme which catalyzes a hydrolysis reaction. A. Lipase. B. Hydrolase. C. Maltase. D. Pepsin. And the answer is B. Hydrolase. pertaining to a group of enzymes that break down carbohydrates. A. Specific. B. Catalyst. C. Lipolytic. D. Amylolytic. And the answer is D. Amylolytic. Any enzyme produced in the stomach and breaks down protein to polypeptide. A. Lipase. B. Gastric protease. C. Maltase. D. Sucrase. And the answer is B. Gastric protease. A method used to manufacture an exact quantity of product, and then the process is repeated for the next consignment. The organism being grown are in a 5-phase growth curve. A. Batch bioprocessing. B. Lytic enzyme. C. Flow bioprocessing. D. Amylolytic. And the answer is A. Batch bioprocessing. An enzyme produced by a small intestine converts peptides to amino acids during the process of digestion. Its optimum pH is slightly basic. A. Lactase. B. Rennin. C. Erepsin. D. Pepsinogen. And the answer is C. Erepsin. Using living organisms to produce useful products, e.g., yeast to produce bread and alcohol, and immobilized enzymes for the production of clear fluid juice, etc. A. Bioreactor. B. Lipolytic. C. Bioprocessing. D. Amylolytic. And the answer is BC. Bioprocessing. The combination of the substrate molecule and the enzyme at the active site prior to their separation and production of the product. A. RNA polymerase. B. Enzyme-substrate complex. C. Bioprocessing. D. Immobilized enzyme. And the answer is B. Enzyme-substrate complex. A container in which a living thing is used in the production of something useful, e.g., during fermentation using yeast, yogurt production using bacteria, etc. A. Bioprocessing. B. Denaturation. C. Lysozyme. D. Bioreactor. And the answer is D. Bioreactor. Proteins with a definitive folded shape. They are highly specific organic catalysts and are affected by heat or temperature, pH, substrate, and product concentration. They control the release of energy in respiration and are essential in the energy transfer process of photosynthesis. A. Substrate. B. Enzymes. C. Immobilized enzymes. D. Denatured. And the answer is B. Enzymes. To speed up a chemical reaction process or to produce something by using a catalyst. This usually involves an enzyme in biology. A. Concentration. B. Denaturation. C. Catalyze. D. Immobilization. And the answer is C. Catalyze. Changes in the shape of an enzyme due to extreme conditions such as high temperature, unsuitable pH, or the presence of certain chemicals. If these changes are permanent, they destroy the activity of the enzyme, and the enzyme is A. Complemented. B. Bioprocessed. D. Denatured. D. Immobilized. And the answer is C. Denatured. Substance used to speed up or slow down a chemical reaction but does not get used up in the reaction. A. Catalyst. B. Substrate. C. Specific. D. Bioreactor. And the answer is A. Catalyst. A method used to manufacture a product without interruption. The organism being grown are maintained in a particular phase of the growth curve, i.e., the log phase. A. Catalyst. B. Immobilization. C. denaturation. D. Continuous flow bioprocessing. And the answer is D. Continuous flow bioprocessing. DNA made from a strand of RNA. Retroviruses produce it for replication, and in biotechnology, using the enzyme reverse transcriptase. A. Transfer RNA. B. Complementary DNA. C. Messenger RNA. D. RNA polymerase. And the answer is B. Complementary DNA. Hello friends, thank you very much for participating in Enzymes. Please do like, share, and subscribe. I'm Joshua Mugabi. Bye bye for now.